Welcome, everyone. Thank you for coming for the second theme of the Magee-Womens Summit, which is The World Within Us, focusing on the microbiome and lifelong health. So it's my absolute pleasure to introduce our first speaker, which is Dr. Elinav, who is a renowned immunologist at the Weizmann Institute for Science, who's really done some of the most groundbreaking work as it relates to the communication and interaction between the host and our microbiota. Particularly, Dr. Elinav has pioneered the field of precision nutrition and using the microbiome as a readout of how we can make best predicted efforts for things like metabolic syndrome and so on. So I don't want to take too much time. Please welcome Dr. Elinav.
Hi, good afternoon, and I'm very happy to be here in this really interesting meeting, and thank you, Yoel, for inviting me. And in the next 30 minutes or so, I'll be kind of speaking to you about the topic of my research, the microbiome. Now, they say that a photo is worth 1,000 words, so I chose to start with this electron microscopy, scanning electron microscopy photo taken at a magnification of 160,000. You can see these bumpy hills that represent the lining epithelial layer of your healthy intestine, and above it you can see what looks like a carpet of shoelaces. So this is your healthy microbiome, and I can already, I think we can already appreciate how extensive this ecosystem that lives within our body from the moment we are born until the moment we die is, and how intimately it is connected to the host side of what we call today the holobiont. And what we do in my two labs, one at the Weizmann Institute of Science in Israel and the other in the German Research Cancer Center in Heidelberg, Germany, is trying to study the microbiome in a highly mechanistic manner in trying to contribute towards better understanding of host microbiome interactions in health and what goes wrong when these interactions or communication networks are perturbed that leads to a risk of developing a number of multifactorial human diseases, including the diseases you're interested in. And by doing so, we and a very large community of researchers around us are slowly but surely contributing towards new principles that I hope and believe would be leading into precision microbiome-mediated disease treatment. And in the next few minutes, I will give you 3 vignettes of our own work that would exemplify where I hope and believe we're going in trying to deeply study the microbiome and reach causality towards the utilization of the immense data that it stores in human disease. Now, when we talk about microbiome-associated disease in 2024, I think it is fair to say that most of the currently used treatment approaches that have something to do with the microbiome are, to put it gently, not sufficiently evidence-based. And we're mainly talking about 3 avenues of research. One is called prebiotics, which are these nutritional, fiber-based interventions that are aimed to make our microbiome healthier, whatever that means. I personally don't know. Fecal microbiome transplantation, which is a method by which you transplant a healthy person's microbial ecosystem into a sick person's microbial system, has been proven to be extremely helpful in several hospital-acquired infections, such as C. diff colitis, but we know much less about its potential roles in more complicated microbiome scenarios. And the third are probiotics, which don't get me started on that, okay? But the more encouraging part of our growing field is that with the immense amount of knowledge and data that is collected about the microbiome, we're now slowly but surely able to gain towards more science-driven and evidence-based experimental microbiome treatment approaches. And again, I will highlight 3 examples, 3 vignettes, but there are many more coming from many more groups around the world. And the first is called personalized nutrition or precision nutrition, which is our version of prebiotic therapy. And this journey started over a decade and a half ago when we realized, really at the beginning of the microbiome field, around 2008, that of the many environmental factors that impact our gut microbiome, nutrition, diet is by far the most dominant and the most important. But so I wanted to start reading the literature that forms the basis of everything that you and I do in our daily lives, usually intuitively, when we try to come up with a healthy diet for ourselves. And when I did this, I read quite boring literature, mainly from the 1970s, and I realized that everything that we do is based on several grading or scoring systems that give different numbers to foods. For example, calories is one such scoring system. And even more widely used grading system for food is called the glycemic index. I'm sure many of you have heard of it. So what is the glycemic index? It's based on very small-scale human trials from the 1970s in which 10 healthy human volunteers were kindly asked to consume an identical food, and then their blood sugar level spikes were measured for 2 hours after they ate that food. So if you were to take 10 healthy individuals, kindly ask them to eat an identical piece of celery, measure their blood sugar levels, you'll see this small average black spice, you would give it a number, and this is the glycemic index of celery. Now, if you were to take the same 10 healthy individuals and kindly ask them to consume an identical piece of chocolate cake, in average, they would spike their blood sugar more, just like the blue graph. You would give this a higher number, and this would be the glycemic index of chocolate cake. Now, take my word for it. If you look in your smartphones, you'll find endless tables that give glycemic index values to any food on Earth. And almost anything that you and I do in our nutritional attempts, whether we go to a physician, to a dietitian, or buy a book at the airport, is based in one way or another on this glycemic index system. But here comes the big problem. When we revisited this using more modern approaches, we found that this grading system is inherently flawed. So the eureka moment came in an early study in which we studied the effects of very commonly consumed nutritional supplements and non-nutritive sweeteners, you know, those sweetening compounds that I'm sure are somewhere on these tables. And their effects on the human body. There's a big argument unrelated to us whether they're inert or not inert. And our kind of 2 cents was that maybe they're inert to the human body, but not inert to the gut microbiome. And to make a very long story short, we gave them to mice and to humans. And what we saw, for example, in this cartoon, is that when you give an identical amount of saccharin, for example, half the people would spike their sugar levels and half the people wouldn't care. Nobody benefited from it. Meaning that the glycemic index cannot tell us about the individuals. We need to kind of give numbers to the individuals rather than to give numbers to the food. This was my eureka moment. So this was the first example. So you know, we expanded it, and we did this in a much larger cohort of 800 individuals, and we gave them, for example, one of several test foods, an identical piece of bread, 50 grams of glucose. We measured all of them. And again, we found that the average glycemic response of any individual to a given food is exactly the glycemic index of that food. That's absolutely correct. But when you looked at the individualized response, there is a huge variation, which means that rather than measuring the foods, we need to find a way to measure the individuals who eat the food. And this was true for the test foods. It was even truer for real-life foods. We've done this on over 100,000 people by now. So this gave rise to a very ambitious project, which we termed the Personalized Nutrition Project, in which we took these 800 or 900 people, and we kindly asked them to give us a week of their life, and we've measured everything that we could measure, the microbiome, because this is what we do in life, but also many questionnaires, dietary information, a continuous glucose monitor, which measured their blood sugar levels every 5 minutes for an entire week, and so on and so forth. And then a very smart group of students took all of this data from each individual and used AI or machine learning technologies in order to devise predictors that would predict a person's response to any given food, including foods they've never consumed during the 1 week follow-up. And now I'm summarizing 6 years of work in one slide, which is a little bit unfair, but it is what it is. And you can see on the left graph the predictability of what we do today. When we go to our physician and we try to reduce our blood sugar levels, we're usually prescribed a diet that is low in carbohydrates all across the board. It has some utility, but the predictability is very low in an individualized manner. In the middle, you can see what happens when you use a person-specific data. You can dramatically improve your predictability. And on the right, you can see a drill that is mandatory in this machine learning predictor development. At some point, you have to stop training your algorithm, freeze it, and apply it to a fresh cohort of individuals who are not part of the training to make sure that you're not overfitting. And we were happy to see that this was the case. So we were mildly pleased with ourselves, but it was time to put ourselves to the real-life test. So we assembled a group of individuals, pre-diabetic individuals. These are people already prone to develop diabetes. Just to give you numbers on the alarmness of this pre-diabetes epidemic, 40% of the adult US population are pre-diabetic. Most of them don't know. They have an 80% chance of developing diabetes within the next decade of their life. So we took this group of individuals, we put them through our system, and then we asked them to eat their predicted good or bad foods for a week while we extensively followed them. And this was completely counterintuitive. A person could be prescribed a little bit of chocolate as their good food and sushi as their bad food. Until you measured, you didn't know. And to make a long story short, we found that even a week of this personalized, counterintuitive good intervention improved their blood sugar levels, and a bad intervention deteriorated. And then when we revisited the algorithm, which is completely unbiased, it doesn't ask for my permission on which feature to use, and saw what it actually used, we were happy to find that at least a big chunk of the information came from the microbiome. So a combination of host and microbiome features were the ones that were driving this good predictability. And so then came the billion-dollar question, which is, can we do something with it over a long period of time? So we generated a randomized controlled trial, 200 individuals, again, pre-diabetic, randomized to receive either the best of the best Mediterranean diet for a year or our algorithm diet while we followed them. And the results were quite striking. You can see in red, these are three different readouts of blood sugar control. So you see in red that the gold standard diet does work. This has been extensively published. It reduces the glycemic indices in these three different readouts. But you can also see that the personalized data-driven approach outperforms the gold standard one-size-fits-all system by quite a large margin. And so this gave rise to a new subfield, which is the precision nutrition. There's many efforts by us and by many others. There's an NIH initiative now in trying to understand the foundations of this and maybe to help people improve their metabolic health. So this is just an example of how you can rationally use data coming from the microbiome in the context of nutrition in order to try and generate rational treatments. So the second vignette that I'm going to share with you relates to the transfer of microbes. I've mentioned fecal microbiome transplantation, which was a very important proof of concept for us back in 2013, that the microbiome stores a lot of potential power to transform human lives. For example, in C. difficile colitis, which is the most common US hospital-acquired infection. And the example I want to provide to you today relates to your, I think, focus of interest, which is another microbiome, the vaginal microbiome, which we developed an interest with. And this is a simpler microbiome than the gut, which is good. And in health, as you know, it is usually dominated by acid-secreting lactobacilli strain. But in a very common female human disease, this lactobacilli-dominated microbiome is replaced by an anaerobic-dominated microbiome, leading to a condition called bacterial vaginosis. This is actually the most common, I think, the most common infectious disease in the world, or one of the most common infectious diseases in the world. Many women, mainly from lower socioeconomic status, but not only, would develop at least one such episode. It would usually be treated with antibiotics, but an amazingly large minority of 4 to 10% of all women worldwide would eventually develop a more stubborn and a form of this disease called intractable or recurrent bacterial vaginosis, which becomes antibiotic resistant. So we're talking about a huge disease, an unmet need, and like many women's related disorders, it is understudied. And the idea, and the important thing is that in this minority of hundreds of millions of women, this disease is not only manifesting as relational discomfort and discharge and so on and so forth, which are important by themselves, but also by a dramatic increase in the rate of complications related to a variety of obstetric and gynecological disorders. So our very naive thought was that maybe just like fecal microbiome transplantation would so nicely impact hospital-acquired gut infections like C. difficile, maybe a vaginal microbiome transplantation could reset the vaginal microbiome and treat or prevent this disease. So in collaboration with an amazing group of OBGYN specialists in Israel, we've worked for many years in developing vaginal microbiome transplantation, making sure that to the best of our ability it is safe and hopefully effective. When we were ready to give it a try, we performed a first-in-human clinical trial in which we took a group of women suffering from intractable bacterial vaginosis for decades without any treatment, and transferring into them vaginal microbiome from a few very healthy volunteers. And the results were quite impressive. You can see here a graph in which on the y-axis you see the clinical score. It's called Amsel criteria, and you can see that all of these women spike to the roof every few weeks with a bout of BV. And once you successfully implant a healthy vaginal microbiome into them, they went into remission for years, and they're actually followed until this day and basically are cured. And what was even more interesting for us was what we saw when we sequenced their vaginal microbiome. So here you can see what we call a principal component analysis graph, which is a way to reduce the dimensionality of the very complex microbiome into a point in an XY axis. And you can see the three colorful full dots on the left, which represent three of the healthy donors. And you can see that their dots are very close to each other, meaning that their healthy vaginal microbiome is very similar to one another, full of good lactobacilli. You can see the red dots, which are very further apart, meaning that these 5 intractable bacterial vaginosis patients have very different microbiomes than those of the healthy controls. But what was most interesting was what happened following a VMT, a vaginal microbiome transplantation. You can see the arrows leading from the original microbiome into a new configuration, which was very similar to the healthy vaginal microbiome. You can even put the clinical manifestations and the microbiome changes on a timely scale. You can see in red the clinical severity score of bacterial vaginosis. You can see that in all of these cases it starts from the roof and it reduces to a healthy configuration. And in perfect timely fashion, the microbiome changes in the reverse way. So it's like a switch. You change the microbiome, you change the clinical manifestations. And here you can see the composition. You can see on the bottom right the 3 donors with this light blue lactobacillus-dominated configuration. You can see these 5 patients starting from a different configuration and moving into the lactobacillus. So this was just an example of how rational bacterial transfer could potentially lead into treatment. What we're doing now is extensively deepening our journey into trying to understand this phenomenon using a large, now randomized controlled clinical trial with VMT, more patients, more configurations, trying to understand what the healthy core vaginal microbiome is. Maybe we can avoid having to do these painful transfers and generate a consortium of microbes, understanding why some women relapse and maybe prevent it, and so on and so forth. And the final vignette that I'm going to share with you today relates to one of our field's biggest unmet needs. And this unmet need relates to the question of, let's say that you become a microbiome researcher and you do your thing and you come up with a microbe that is strongly associated or even contributes to your favorite disease, whatever it is. How do you get rid of this bad microbe? And if you think about it, there's 0 solutions to getting rid of a microbe without impacting the entire ecosystem that surrounds it. So antibiotics are a very bad way to do this. They're non-specific. They're associated with the emergence of resistant strains, with adverse effects. You cannot give antibiotics forever. So we really had no way of doing it. So 3 champion postdocs at the lab 7 or 8 years ago decided to try and tackle this huge unmet need. We decided to focus on a disease called inflammatory bowel disease, a very microbiome-associated disease, which we study a lot. And in order to develop such targeted therapy, we first had to figure out which microbe to target, right? So we generated a large cohort in 4 different countries, in France, in Israel, in US, and in Germany. In each of these, we had IBD patients, both ulcerative colitis and Crohn's disease. These are the 2 subtypes of IBD, and healthy controls in the same country. And this is a very important paradigm for us because the microbiome is exquisitely prone to being affected by ethnicity, by diet, by lifestyle-related features. So doing a study in different countries and able to optimize the chances of finding generalizable signals that relate to a disease across different populations. So we were happy to find what was shown before us, that the microbiome in IBD patients as a whole group is different as compared to healthy controls, both as an entire group on the left and in each of the countries on the right. So the overall premise was correct. And given the fact that we've done this in so many sites and sequenced the microbiome very comprehensively by shotgun metagenomic sequencing, we were able to generate a short list of commensals that are altered in IBD patients across the different cohorts in the different countries. And this is your short list. And since we had to start somewhere, we decided to focus on the top hit, which is Klebsiella pneumonia, which was expanded in all the IBD patients in all the countries, and was even expanded in flaring patients as compared to patients in remission. So this is by no means the only bug that is correlated to IBD, but it was the bug that we decided to focus on. And since we're great believers in going from even species level to a strain level specificity, we generated a computational pipeline that enabled us to look into the strains of Klebsiella pneumonia, and we were surprised to find that many of these IBD-associated human KP, Klebsiella pneumonia strains, came from a specific subgroup of Klebsiellas, from a specific clade of strains that were closely associated with each other. So our big aim was not only to target Klebsiella pneumonia as a species, but to actually target this subgroup of Klebsiella pneumonia that is so associated with IBD. But before that, we wanted to go from correlation to causation, which is one of our biggest missions in life. And to do this, we fished out from these hundreds of patients, hundreds of different KP2 or not KP2 strains. We call this clade of IBD-associated Klebsiella pneumonia KP2 and all the others non-KP2. So we fished them out, we cultured them out, and then we used germ-free mice. These are mice that are housed in specialized isolators and don't have any microbiomes of their own as recipients of these different strains to see what the different strains would do to a disease model simulating IBD in mice. And for the sake of time, I'll just show you 2 short examples of these causality-seeking experiments. For example, in the first, we've used germ-free mice that report an anti-inflammatory cytokine called IL-10. So whenever it is secreted, we know of it. And then we put into them, one by one, all of these KP2 or non-KP2 human strains. We measured the different pro- and anti-inflammatory cytokines, and what we found was that the KP2 strains were inducing a very pro-inflammatory Th1 response, which is hallmarked by low IL-10 secretion and high interferon gamma secretion, which is also a hallmark of some subsets of human IBD. In a second example, we used a different type of germ-free mice. These are mice that are missing this anti-inflammatory cytokine. So these are mice that are prone to spontaneously develop IBD. And then we put into them the different strains. And what we found was that the KP2 strains, when put into this genetically susceptible IBD-prone mice, induce a very severe form of IBD. You can see in the middle panel one of the biomarkers, the pro-inflammatory biomarkers that goes to the roof upon this experiment. Again, you can see that the T cells secrete lots of interferon gamma. upon this colonization, and the result is a huge inflammation and tissue damage, which we quantified by histopathology, as you can see here. So at least in these experiments, we had a preclinical evidence that KP2 is not only associated with IBD, it is actually contributing to IBD. So now came the billion-dollar question: what do you do with it? How do you get rid of these strains of KP2? And what we chose to utilize are cute viruses, which are called bacteriophages, which we like a lot. And we like them because these are phages that only target bacteria. Their big enemy in life is bacteria, and they go after bacteria. There are no known eukaryotic receptors for phages, and therefore they are perceived as being very safe to humans or to mammals. And the idea was that maybe we can find phages that target very specifically Klebsiella pneumonia or these strains, suppress these strains while not impacting the surrounding ecosystem, and this would be a targeted therapy. So why this could be a good form of targeted microbiome treatment? Because these phages are very specific. They attack the bacteria through a set of receptors. They have a narrow range of specificity. They've been shown to be non-toxic because they cannot infect humans. In fact, they've been extensively used as a cultural kind of treatment in the former Soviet Union because there was no money and availability of antibiotics for many years. So phages, for some reason, became very popular. You can still buy them in local supermarkets. The science was extremely bad, no IRB, no randomized controlled trials, but there's a lot of data on safety. So they are very safe, and therefore they're recognized to be safe. So phages have been known for 100 years. Why haven't this been done before? Two big challenges, actually huge challenges. First of all, if you give phages systemically, phages are composed of many foreign proteins. They would work in the first time you give them, in the second time an immune response would eliminate them. And there are many case reports of people suffering from antibiotic-resistant infection and phages being used as salvage therapy. Always the response is a short-term response. The way we wanted to solve this challenge is to give phages orally, because we wanted to target the gut microbiome and thereby avoid the bulk of the immune response. The even bigger challenge is what we call phage resistance. So if you were to give a single phage to a bacterial colony, there's a 100% chance that one of the copies of the bacteria would be mutated for the receptor to the phage, and it will take over, and you will have phage resistance. In addition, if you read the big basic science literature in the last couple of years, you would learn that bacteria and phages are the arch-rival enemies of each other, and therefore in evolution they've developed ways of protecting themselves from each other. So there is a bacterial immune system against phages. For example, CRISPR is a bacterial adaptive immune system against their enemy phages. Restriction enzymes is another way by which bacteria protect themselves from phages. And there are many, many new systems that are being discovered as we speak. So how do we solve this? We decided to try and generate phages that attack Klebsiella pneumonia through different receptors, to generate a cocktail of phages that would be given together, just like an HIV cocktail or any other cancer cocktail, and the bacteria would not be able to develop resistance against all these different phages. So this was easier said than done, and in reality what it caused is my students and postdocs to go with buckets to the Israeli sewer system, take the stuff and isolate phages and sequence them and look for specificity. So we've done this for quite a long time. This generated very boring Excel sheets with many phages isolated, resistant, sensitive. And at the end of the painful process, we identified 49 phages that target Klebsiella pneumonia, specifically by in vitro culturing. It seemed to do this against KP2. And we took these 49 phages and we put them into groups, into cocktails of different combinations. And again, we performed a series of in vitro cultures using as their target the most resistant KP2 strains of bacteria that we had at our disposal. And this generated many boring growth curves to the different cocktails. And we ended up with 5 cocktails that reached the semifinal. So these were cocktails that seemed to be effective at the in vitro level against all the Klebsiella, the KP2 strains that we co-cultured them with, even the most resilient ones. So this gave rise to the final stage, which is the in vivo stage. We took these 5 finalist phage cocktails, and we applied them into mice that were colonized with a very evil KP2 strains that we had at our disposal to see whether some of these cocktails would be doing the same thing at the in vivo setting, which resulted in another large group of growth curves. And this gave rise to the grand winner, a cocktail called 5E. And I'm sure you're dying to meet your winners. So here they are. These are 5 phages. Of course, we sequenced them. They come from 5 different families as expected. Now our big test was to prove that this cocktail is actually not just able to suppress Klebsiella, but actually able to suppress IBD, at least in the models. So we went back to the mice, and we did many experiments. I'm just going to show you a couple. You can see here mice that are colonized with a very resistant IBD-causing KP2, and now you apply on top of that the 5-phage combination. You can see on the right bar, in the second lane, you can see that when you just give the KP2, it induces this huge inflammatory response. I've shown it to you before. And now when you put on top of it the phage cocktail, it not only suppresses the bacterium, it suppresses the immune response in the host mouse. And now you can induce full-blown IBD or a model of IBD in mice. You can see that the phage cocktail reduces the load of Klebsiella pneumonia, both in the lumen in the left and in the mucosa on the right. And this induces a suppression of the immune response. These are examples of cytokines that are suppressed by the phage cocktail. And the resultant tissue damage is attenuated. We measure this by murine colonoscopy and by histopathology. And this gave us a proof of concept that these phages could actually suppress Klebsiella, and through this suppression, suppress or attenuate the severity of IBD models in mice. And even when we gave phages in chronic models of mouse colitis or mouse IBD models, we found no evidence for the emergence of phage resistance because of the cocktail. So the suppression was long-lasting, at least for a few weeks. And under these long-lasting conditions, we could even observe a survival advantage of mice that were induced with this chronic IBD and were treated with the phage cocktail. And finally, we wanted to scrape the surface and go back to humans and see whether this treatment at least would be plausible as a potential human therapeutic. So we performed a first-in-human clinical trial, taking or recruiting a group of healthy volunteers and giving them either phage cocktail or vehicle control and following them for around a week. What was really special about this trial, in contrast to everything that we do in humans, is that we didn't just sample their fecal samples. We actually collected everything that they had to offer during the trial period, which reflected on our -80 refrigerators, but it also enabled us to very accurately assess the total quantities of the virus in their human gastrointestinal tract and to see whether the viruses thrive and proliferated in the human GI tract. And the answer was a strong yes, and we could identify that the viruses were very happy in the lower GI tract of these healthy individuals. They actually reached concentrations that were 1,000-fold higher than what would be expected for them to suppress Klebsiella pneumonia. Of course, in this first in-human trial, we could not quantify the suppression of KP2 because these were healthy individuals, and I started by telling you that KP2 is enriched in IBD patients, so this awaits the phase 2, which we're almost underway. But what we could assess is the possible off-target effects, whether these KP2 targeting phages were doing anything to the many other microbiome commensals that were around them. And of course, we sequenced these individuals very extensively, and our answer was no. There is actually no effect on the general composition of the microbiome or on the abundances of the strains surrounding, but unrelated to the Klebsiella pneumonia that these phage targeted. And this gave rise to a lot of interest, and we've formed a spin-off company, Biomics, which further goes after other bugs. For example, Pseudomonas aeruginosa in cystic fibrosis patients, which is a huge problem of antibiotic resistance. So they've generated a different cocktail and tested it successfully in pulmonary inhalation. There are other groups that now are developing other cocktails of phages against other pathobionts or commensals that are contributing to human disease. So this may be one such solution that is worth following and exploring. And to summarize my talk from a bird's eye view, I just gave you a few examples of data-driven or science-driven approaches by which we try to increasingly utilize the microbiome towards the development of rational therapies that I hope and believe would be incorporated into the precision medicine shelf within the next decade of research. And I'll finish by thanking the amazing people that have joined me in this journey from all over the world at my lab at the Weizmann Institute of Science, the many clinical and preclinical collaborators without whom we couldn't have done anything, the funding agencies that are making our work possible. And for the past 5 years, I've engaged in a parallel adventure by establishing a unique microbiome and cancer division at the German National Cancer Center in Heidelberg. A fantastic group of people as well, and what we do there is try to apply the technologies that we've developed at the Weizmann to study whether the microbiome may impact the development, progression, and response to treatment in the cancer context. And with that, I think I'll stop here. Thanks.